Hi, today we are here with Brian O'Dwyer of ICON, the Senior VP of Global Bioanalytical Services. Welcome, Brian.
Hi, Elizabeth.
We're going to talk about two trends in drug development today, one being biosimilars and the other biomarkers. Starting with biomarkers, what is ICON doing in this space to increase the use in drug development programs?
Well, we recently announced that we've done some lab expansions in our UK and US laboratories in our research and our diagnostic biomarker space. So we've now got a dedicated research biomarker lab in the US to mirror the one that we have in our European labs. We've also recently invested in newer technologies by bringing on board the Randox evidence multiplexing platform and by adding our fourth GyroLab station in the US, which makes us having 4 GyroLabs globally, which makes us the largest CRO using gyros in the world. As a contract laboratory working in the biomarker space, it's very important that we offer a range of platforms to our sponsors, because depending upon the drug, its mechanism of action, as well as the biomarker target that the client is looking for, the number of analytes, we have to be able to offer a range of platforms. So by adding new platforms, we're able to offer that. And the nature of biomarker testing is that each assay has got to be developed and validated on a fit-for-purpose basis to meet the need of that development. So we need to have very high scientific capabilities in our leadership team, so we keep adding that into our group as well.
What is ICON doing bioanalytically to support biosimilar development?
So early in 2012, ICON decided that it would develop its own assays internally for the leading biologics that are coming off patent over the coming years. So we've developed our own PK, immunogenicity and neutralizing antibody assays in line with the current regulatory guidance that has been recently issued, as well as in consultation with our clients who are developing these compounds. The approach that we've taken is that we are developing a single assay for all PK testing, a single assay for neutralizing antibody testing using either a cell-based assay or a plate-based assay. And we've developed an individual assay for the immunogenicity test for each of the molecules, both the European compound, the US compound, and the biosimilar that's under development. We believe that that's the approach that the regulators are going to want to see. And we also take a statistical analysis approach in determining that the assays are equivalent in the assay. So what's the benefit of that to the industry? Well, by us internally developing these assays, it means it will save our sponsors the lead time in getting the assay spun up and ready to go for testing, as well as saving them cost. So it's a more cost-effective and efficient way for them to develop a biosimilar using our bioanalytical labs.
Thank you, Brian. To learn more about this, please visit www.iconplc.com.